All right, let's dive in. Switzerland, late 1800s, early 1900s.
And son.
You sent in this academic paper, The Beginnings of Developmental Biology in Swiss Universities.
Oh, yeah.
It's amazing. So many pioneers figuring out how creatures develop.
Embryos, sea urchins.
Newts, even using UV light to make sterile flies.
Absolutely. It's fascinating how this period shifted the whole field, moving from observation to experimentation.
Yeah, let's start with Wilhelm.
Okay.
The paper calls him the guy who revolutionized how we study embryos. Darwin's theory of evolution was huge then, got scientists thinking about development in new ways.
Yeah, his really saw descriptive embryology as the key, meticulously documenting each stage, showing how each is a consequence of the one before.
So not just what was happening, but how and why.
Exactly. And his methods were so innovative for the time. He sliced embryos super thin, like a microscopic deli slicer, then reconstructed them in 3D.
Wow, the 3D puzzle, but tiny?
Right. He also came up with this idea of organ-forming regions in chick embryos. Specific regions destined to become certain organs. It was the groundwork for later techniques like fate mapping.
Wow, way ahead of his time. This was all before any fancy molecular techniques, right? Just observation and intuition.
Totally. And he made big contributions to histogenesis, too, how cells form tissues.
The building blocks.
Exactly. And he even contributed to the neuron theory that the nervous system is made of individual nerve cells connected together.
So broad impact, not just on embryology.
Right.
The paper also mentions he wasn't a fan of Haeckel's famous biogenetic law illustrations, the embryos going through stages that look like their evolutionary ancestors.
Yes. His Pratenge challenged those illustrations. Based on his detailed work, he thought they were oversimplified and inaccurate. Turned out he was right.
They were debunked.
Exactly. It reminds us that even in science, things aren't always what they seem.
His work really set the stage for the next generation. Guys like Fritz Balter, who started using experiments.
Yes. Balter was a student of Theodore Boveri, one of the scientists who developed the chromosomal theory of inheritance.
That chromosomes carry the genetic information passed down.
Yes. And Balter brought this new understanding of genetics to his work with embryos. He was fascinated by how instructions in the chromosomes interacted with the rest of the cell.
It sounds like he really launched experimental embryology in Switzerland. What was he doing with sea urchin hybrids?
Oh, that was fascinating. Crossing different species, looking at their embryo development. It was tricky back then, but he created these hybrid embryos. He observed that in some cases, chromosomes from the father's sperm were actually eliminated during cell division.
Wait, so some genetic instructions were just disappearing?
Yes, and it impacted how the embryo developed. It was a big finding because it supported Boveri's idea that each chromosome carries unique, essential information.
So not just the number of chromosomes, but the right instructions from both parents.
Precisely. Development depended on the interplay between genes and the cellular environment. Not enough to have the blueprint, you need the construction crew to interpret it.
Like a conductor in orchestra. The conductor has the score, but the musicians bring it to life.
Perfect analogy. And Balter went on to study this worm Bonellia viridis. They have this crazy sex determination system. Females are large, but males are tiny and live inside the female.
Wait, the males live inside the females? How?
I know, wild, right? Balzer figured out it all came down to environment. Larvae in water became females, but if a larva touched a female, it developed into a tiny male living inside her.
So the environment dictated their sex.
Exactly. And even the time near a female determined if it became a fully functional male or something in between intersex. Groundbreaking stuff.
He was showing that environment could shape you, at least for these worms.
Challenging the idea that it was all about genes, external factors had a huge impact.
Okay, so sea urchins, Bonellia viridis, that's just the beginning. He also did amazing work with newt embryos, right?
Oh, yes. He was a master at manipulating embryos. He created embryos with half the usual number of chromosomes, called the marigones.
How did you do that?
Very carefully. He would manipulate newly fertilized eggs, separating them into fragments. Some contained only the sperm nucleus. Others had the egg's nucleus and a few sperm nuclei. He was creating haploid embryos.
Haploid meaning a single set of chromosomes instead of the usual two.
Exactly. He wanted to see if a single set was enough for normal development. And sometimes it was. But when he combined egg cytoplasm from one newt species with a sperm nucleus from another, things got messy.
So not just a number, but also where they came from.
You got it. The success of these hybrid marigones depended on how closely related the species were. If they were distant, development was abnormal, like the genes and cellular environment had to be speaking the same language.
Like building a house using blueprints from another country. Wrong measurements, incompatible materials, wonky house.
Haha, exactly. Marigons highlighted the complex dance between genes and the environment. But he didn't stop there. He wanted to understand how cells from different species interacted, so he used transplantation to create chimeras.
Chimeras, like the mythical creatures.
Yes, but in this case, embryos with cells from different species. For example, he transplanted head neural crest cells from a toad into a newt embryo. Neural crest cells give rise to many different tissues in the head.
So mixing and matching embryo parts.
In a way, yes. He found the toad cells helped build the newt's visceral skeleton, part of the skull. But the structures had mixed toad and newt characteristics. They cooperated, but not perfectly.
Like a Lego castle using pieces from different sets. Might fit, but looks a little off.
Exactly. He concluded that the level of cooperation reflected how closely related the species were. The closer, the better they worked together. His work showed that development was deeply rooted in evolutionary history.
Incredible. Balzer was a master. He really took embryology to another level.
He did. His work paved the way for the next generation of Swiss developmental biologists, like Fritz Erich Lehmann. He delved even deeper into the chemical side of development.
That sounds like a story for next time.
It is. Lehmann's fascinating work in part 2 of our deep dive. Stay tuned.
Back again, diving deeper into these Swiss developmental biologists.
Picking up where we left off.
Last time was Fritz Balzer.
Oh, yeah. A master manipulator of embryos.
Now it's Fritz Erich Lehmann's turn. Started in medicine, then switched to zoology.
Quite a change.
A good one, it seems. Big for developmental biology. He focused on insects for his thesis, right?
He did, and he had some pretty impressive mentors, Balzer, but also Hans Spemann in Germany, Ross Harrison in the U.S.
Well, the all-star team of mentors. No wonder he went on to do some groundbreaking work. What kind of research was he into?
Well, Lehmann was all about the chemicals, how they influenced an embryo's development, the chemical basis of development.
This is where chemical embryology comes in.
Exactly. He's considered a pioneer. He was really interested in how chemicals affected morphogenesis, the process of an organism developing its shape and structure. One of his key studies involved lithium chloride.
Libre. Like in batteries.
The same. Sounds strange. But he found that exposing newt embryos to lipiro caused specific birth defects.
So he was disrupting the normal process with chemicals.
Exactly. And the type of defect depended on when the embryo was exposed and which organ was affected. For instance, Lemurel at a specific stage disrupted the head mesoderm, which forms the brain and sense organs.
Like specific windows of vulnerability. Kind of like building a house. You can't change the foundation once the walls are up.
Perfect analogy. And this wasn't just theoretical. Lehmann's work connects to those real-world tragedies with birth defects from certain drugs.
Thalidomide, given to pregnant women for morning sickness.
That's the one. Sadly, it caused severe birth defects. Lehmann's research showed how chemicals can have very specific effects on development, especially the timing.
Underscoring the importance of those early stages.
Exactly. What happens early can have lifelong consequences. You could say Lehmann's work aimed to understand these developmental mishaps.
To prevent future tragedy.
Yeah. And his research went even further. He was also interested in using chemicals to block cell division and tissue growth, control those basic processes.
Like hitting the pause button on cells multiplying.
He used the regenerating tail tip of Xanapis tadpoles for this. Those African clawed frogs that can regenerate their tails.
Like science fiction.
I know, right? He was looking for chemicals to stop this regeneration, and he found them.
So putting the brakes on cell division and growth?
Yes. And he found that combining different inhibitors, each targeting a different step, was even more effective.
A double whammy. But why block regeneration? That's usually a good thing.
Think about it. Uncontrolled cell division causes cancer. If we can control it, we might be able to treat cancer better.
So implications for fighting cancer. That's huge.
Basic research is so important, you never know where the applications might lead.
Chance favors the prepared mind, as they say.
Exactly. Lehmann was always thinking outside the box. This is clear in his later work on the cytoplasm's role in controlling cell fate.
Wait, the cytoplasm? I thought the nucleus with the DNA was the control center.
It is, but Lehmann wanted to know what role the rest of the cell played, questioning the dogma.
Looking beyond the obvious.
He used cutting-edge tech for the time, electron microscopy, saw the tiny structures inside cells in incredible detail. He was studying stem cells from tubifex embryos, those segmented worms.
Stem cells, the ones that can become all sorts of cell types.
Exactly. And he found that different types of stem cells contained different submicroscopic particles in their cytoplasm.
Visible only with the electron microscope.
Right. And he thought these particles were somehow connected to what a stem cell would become.
That they were influencing the cell's fate.
Bold idea. It wasn't widely accepted back then.
It makes sense, though.
Well, back then, the nucleus and DNA were thought to be the sole dictators of cell fate. Anything else was radical, like saying the construction crew could change the blueprints.
That is a big shift. But those radical ideas can be game changers.
They are. And Lehman's work certainly pushed the boundaries. It set the stage for later discoveries that confirmed the importance of those cytoplasmic factors.
Amazing what they discovered with such limited tools.
Truly, he was a pioneer, challenging the status quo. But there was another Swiss scientist working around the same time, Rudolf Geigy, who also made big contributions. His focus was hormones in development.
Oh, tell me more about Geigy.
Fascinating story. He started as a zoologist, working on both insects and amphibians. Later in life, he actually founded the Swiss Tropical Institute.
From embryos to tropical diseases, that's a leap.
It is. But his early developmental biology work was impactful, especially his research on how UV light affected insect development.
UV light, like sunlight.
Yes. He wanted to see how UV radiation could disrupt development. He had this precise method to expose parts of Drosophila eggs, fruit fly eggs, to UV light. Found that if he hit the posterior pole, the flies would be sterile.
Sterile flies?
Yeah.
Wow, what's so special about that spot?
It's called the polar plasm, and it's vital for developing germ cells, those that become sperm and eggs.
So damaging it with UV light stopped them from producing sperm and eggs.
Exactly. Clever way to show the importance of specific parts of the cytoplasm in early development.
It's incredible how they did these experiments back then, manipulating such tiny structures. They must have had amazing skills and patience.
They were pioneers, pushing the limits, and Geigy didn't stop there. He also studied amphibian metamorphosis, the tadpole to frog transformation.
It's like magic. You start with this fish-like creature, then bam, a frog hopping around.
It is incredible. And Geigy wanted to understand how it was controlled. He used transplantation, moving tissues between tadpoles to see how it affected metamorphosis.
Like tadpole surgery.
Pretty much. Very skilled with those tiny instruments. For example, he took the tail bud from one tadpole and put it on the trunk of another.
Did it grow two tails?
At first it did, an extra tail where he put the transplant. But get this. When it went through metamorphosis and became a frog, both tails disappeared, just like normal.
Wow. So even in a new spot, it followed the same program.
Exactly. He also tried transplanting trunk tissue into the tail bud of another. And during metamorphosis, while the tail tissue around it disappeared, the trunk tissue stayed put and developed into normal trunk structures.
It held on to its identity, even surrounded by tail tissue.
Like it had an inner compass guiding its development. He concluded that the changes during metamorphosis are controlled by properties of the larval tissues themselves. Each tissue has its own program for responding to the hormonal signals that trigger metamorphosis.
So it's not just the hormones calling the shots.
Right. He compared it to a train schedule. The hormone is the train. Each tissue is a passenger getting on at its designated stop.
Love that analogy. But we know that thyroid hormone is the main trigger, right? Did Geigy look into that?
He did. He wanted to know how thyroid hormone worked with those tissue programs. He found that the ability of larval tissues to respond to thyroid hormone, their competence, developed gradually and differently for each organ.
So not every tissue becomes sensitive to it at the same time.
Nope. It's like a choreographed dance. Each tissue responds at the right time, ensuring metamorphosis happens in order.
Amazing what they figured out without today's tools.
I know. Their ingenuity and deep understanding of biology, their work laid the groundwork for how we understand hormonal control of development in amphibians and all vertebrates, including us.
That's what makes these deep dives so cool. Seeing how these discoveries, even from a century ago, are still relevant.
Absolutely. Science is a continuous process, each generation building on what came before.
It's really amazing how much these Swiss scientists figured out. Back in the late 1800s and early 1900s, it's like they wrote the textbook on developmental biology.
True pioneers. They had a knack for taking these big, complex things like metamorphosis and breaking them down into manageable pieces they could actually study. And they were resourceful, using whatever tools they had, a scalpel, a microscope, or even just UV light.
Proof that the simplest tools can sometimes make the biggest difference. We've covered a lot in this deep dive. Embryo reconstructions, those sea urchin and newt experiments, even using UV light to sterilize flies. These guys were really leading the way.
They were at the forefront, shifting developmental biology from just observing to actively experimenting, testing ideas, manipulating embryos to see how they tick.
And they asked some big questions. The role of chromosomes, how environment influences sex determination, even the chemistry behind how an organism gets its shape. It's mind-blowing what they figured out with the tools they had back then.
It is. Imagine if they had today's technology. Gene manipulation, tracking individual cells, analyzing molecular interactions, things they couldn't even dream of.
It's like comparing a horse and buggy to a spaceship. Makes you wonder what amazing discoveries are still out there.
Developmental biology is such an exciting field. There's still so much to uncover, and with all these new technologies, we can ask questions that were impossible before.
It's what's so cool about science. Every answer leads to more questions, and that search for knowledge never ends.
A never-ending journey of exploration and discovery. And these Swiss pioneers, their creativity and curiosity really put us on the right track.
What I love about these deep dives is seeing how these early discoveries, some over 100 years old, still matter today. Shows how crucial basic research is.
Couldn't agree more. It's about that scientific curiosity and exploring the fundamental mysteries of life.
Well, I think we've successfully navigated this deep dive into early Swiss developmental biology. It's been fascinating learning about these pioneers and their work.
It really has. And for our listeners, as you go about your day, think about this. What other scientific fields might be on the verge of big breakthroughs with new technology? The answer might surprise you. Keep those brains buzzing and never stop exploring the wonders of the natural world.
Thanks for joining us on the deep dive. Until next time.